All right, last chapter. So obviously last time we talked about digestion. We talked about breaking down things like carbohydrates, breaking down proteins, lipids, etc., and incorporating them into the body, right? So we talked about absorbing them in the small intestine and moving them into the bloodstream. And in this chapter, we're going to talk about what happens then, what happens next in that process. So it's actually kind of three parts. So there's carbohydrate metabolism, lipid or fat metabolism, and then protein metabolism. So I'm going to break this up into two parts. So we're going to, in this video, cover carbohydrate metabolism, and then in the second video, we'll get to fat and protein metabolism. So before we get to that though, one of the things I've tried to stress over and over in this class is the idea of ATP, which is adenosine triphosphate, as the energy currency, right? And so we use ATP to do a bunch of different things. So for example, back in chapter 9, when we talked about how muscles work, remember that we use ATP, we split its last phosphate off of it, and the energy that's given off whenever that happens, we use that to reset the spring, if you will, to reset the myosin heads so that when they see those binding sites on actin, they can jump up and attach and pull it toward the midline. So we use ATP then to power skeletal muscle contraction. We've talked about sodium-potassium pumps and how we use ATP to pump those ions against their concentration gradients. You can see there from my picture that ATP is used as part of exocytosis during the secretion process of different hormones, right? So we use ATP to do all kinds of different things. But what we have to do then is to use the energy from the macronutrients. So the macronutrients are carbohydrates, fat, and protein, and alcohol. And we use the energy from breaking the bonds in those macronutrients to help us rephosphorylate, to help us reform ATP. So I'm pretty sure we've seen this picture before. It's in the lower right. So ATP, again, adenosine triphosphate. So you got your little adenosine backbone here. And then again, triphosphate, tri is 3. So you have your 3 little phosphate groups. So here's the first one, and then the second little P there, and then the third one there. So those are our 3 little phosphate groups. And what you can see on this picture, this little, I don't know, almost looks like a don't sign. In really close up, it's a little squiggly line. But what these two things are, or what they're meant to represent, are high energy bonds. And so when we split this high energy bond here between the last two phosphates, that gives off free energy. And we use specific enzymes to do that. Those are ATPases. And so, for example, in the muscle cell, that was myosin ATPase that split that last split that last bond and gives off a bunch of free energy. So once we do that, then we're left with an adenosine diphosphate. So you only have 2 phosphates left attached to the backbone. And then you have a little free phosphate. And so what you see over here, so we've got ATP, adenosine triphosphate, and then when we split it, the byproducts there, or the products of splitting it, so you end up with ADP, adenosine diphosphate, and then the little PI is our free phosphate. So those are the products of doing that. So as you can see, this is a high energy bond as well. So we could split that, but we prefer not to do that. So effectively what has to happen then is we need energy in order to push this last phosphate bond back onto the adenosine diphosphate. And so we have to rephosphorylate, remake ATP from a free phosphate and an ADP. And so by breaking down the macronutrients, we're going to use the energy from splitting those bonds in order to help us remake ATP. So one of the ways to think about this process is basically when you think about the macronutrients, so something like carbohydrates. The basic chemical formula for carbohydrates, we'll get to it on a slide here in a second, but it's CHO, so it's carbon, hydrogen, and oxygen. And so effectively what we do during this process is we're going to, in stepwise fashion, pull off pairs of hydrogens off of those molecules, and we're going to use the energy from doing that, from breaking those bonds, again, to help us remake this ATP. So the idea is that obviously a glucose is a pretty big molecule, and so if we broke all the bonds at once, that would give off too much energy, right? And so we wouldn't be able to do any productive work with that. So we have to systematically break a few bonds at a time so that we can harness the little bit of energy that's given off every time we do that. So that's kind of one of the ways of thinking about it. Most books, I think including our anatomy book for this class, use the analogy of being like an engine in a car, that the way that the car is powered is by burning a little bit of gasoline at a time, and obviously the different parts of the engine help convert the energy that's given off when we burn that gasoline into driving the car forward. And so if we broke all the bonds at one time, if we burned all that gasoline at one time, that would cause an explosion, right? And that wouldn't be very useful for powering the car. Same concept here. So that's why we have to incrementally or step by step break those bonds in the macronutrients. So in terms of my actual slide here, so the ATPs split almost instantaneously without the need for oxygen. So it's just one enzyme, it's an ATPase. That enzyme splits that last bond, gives off free energy, and then again that helps us power cellular work. One of the things that occurred to me when I was taking classes where we talked about ATP early in my college career was, okay, so we have all of these processes, whether it's, as we'll see here in a second, it's this combination of glycolysis and the citric acid cycle and the electron transport chain. So we've got all these really complex processes to help us remake ATP. Like, why can't we just have just a boatload of ATP? And the answer is that ATP is a fairly heavy molecule. So to give you a little statistic on that, even a person who is sedentary, if you just sit around all day, they resynthesize an amount of ATP that's equal to about 75% of their body mass throughout the course of the day, right? So if you've got, should have done this math in advance, but if you have somebody who is, I don't know, let's pick an easy number. So you got somebody who's a 200-pound person, sits around doing nothing, the amount of ATP that they resynthesize per day is equivalent to about 150 pounds, right? So it's a fairly heavy molecule. And what that means then is we can't store, we can't carry very much of it. And so things like fat are much more space efficient, if you will, right? They're much lighter. You can store a lot more calories, a lot more energy in a lot less space. And so because of that, it's easier for us to use things like fat or lipids as a storage form and then break those bonds to help us remake ATP than it is for us to store a lot of ATP just because it's so much heavier. Another statistic there, just because it's interesting, that an elite runner in a marathon can regenerate about 175 pounds of ATP over the course of that little more than 2-hour race, right? So that's, for most of your elite runners, that's actually going to be equivalent to more than their body weight worth of ATP throughout the course of a 2-hour or just above 2-hour marathon. So an important thing about that, the last part on there, again, we only have one enzyme involved in splitting that last phosphate off and giving off free energy, so an ATPase of some sort. So one enzyme, split one bond, and we get free energy. So it's really quick. So we've got enough energy stored in the muscles to power a few seconds of energy without the need for oxygen. So if you need to like sprint to get away from something or do something really explosive, like a big jump or a big lift or any of those sort of things, you have that amount of energy stored in the muscles, but just not energy available that quickly for much longer than a few seconds. So that's ATP. So the fastest way to rephosphorylate. So if I phosphorylate something, I add a phosphorus. So when I say rephosphorylate ATP, what I mean is to put that phosphorus group, that last phosphorus, back onto adenosine diphosphate. So the fastest way to rephosphorylate or to remake ATP is to use phosphocreatine. So what you can see here at the bottom is, so the little PCR, that's your phosphocreatine, right? And actually we'll start up here where I should. So you got ATP. We split it using ATPase, right? And then the products that we have are adenosine diphosphate plus free phosphate plus our energy that we gave off. Now, if I want to, so again, myosin ATPase fuels muscle contraction, if you will. So if I want to continue muscle contraction, well, I got to continue my supply or remake my supply of ATP. So again, the fastest way to do that is also in the muscle cell, we're going to have a store of phosphocreatine. And so you have a little phosphate molecule here. And so what we can do then is combine our phosphocreatine and our adenosine diphosphate. The enzyme that's going to catalyze that reaction is creatine kinase. And so what we end up then with is an ATP and then a little free creatine. So The good thing about that is that creatine phosphate can give up its phosphate group. Again, we've only got one enzyme involved in this, and so with one enzyme in one step, we can remake our ATP. So it's a really fast way of remaking ATP. Then that ATP can be split, and then we can get another interaction of actin and myosin, right? So good thing there, really quick reaction, one step. We can actually store much more phosphocreatine than we can ATP because it's not as heavy as ATP, but it is still a fairly heavy molecule. So another thing, or a thing with that, same concept with creatine phosphate. The reason we don't store tons and tons of it is, again, because that's a fairly heavy molecule. So where some of you might have personally noticed that, if you've ever supplemented with something like creatine monohydrate, the typical supplementation scheme is that you load it for a week, right? So you take 20 grams a day for a week, and then for maintenance, you take 5 grams a day for however long you want to use it for. And so when you load it, people often notice that they gain somewhere in the neighborhood, depends on your body size and how much muscle mass you start with, but people typically gain in the neighborhood of 3 to 5 pounds if they, after that loading phase, after that first week of taking 20 grams a day of creatine. And so they've gained 5 pounds, but have they added 5 more pounds of muscle? Well, of course not. But what they've added is, so as the creatine phosphate, or as the creatine becomes phosphorylated inside of the muscle cells, it's going to pull some water with it. And so the muscle cells swell. And so then that water retention is what produces the initial gains in size. Now, don't have time to talk about the benefits of creatine supplementation for your strength and power athletes. It helps recovery between sets. It helps recovery between workouts. Helps you maintain power outputs for longer. So it does a number of different really beneficial things for strength and power athletes. But again, because it causes water retention, it's a fairly heavy molecule. So we can only store enough creatine phosphate to power muscle contraction for an all-out bout of about 10 seconds. So oftentimes you'll hear the two systems referred to as the ATP phosphocreatine system. So between those two systems, again, you have enough fuel to power all-out muscle contraction for about 10 seconds. So for about enough time to run about 100 meters or a little bit less, because most of us aren't really that fast. So, 80 meters, 100 meters, somewhere in there. And then after that, you've pretty much exhausted the ATP phosphocreatine or your phosphocreatine stores. So we have to use something else to help us remake our ATP. And so that's where carbohydrates come into this equation. So that... and again, from a power standpoint, one of the ways to conceptualize all of this stuff is the fewer steps that are involved in remaking ATP, the faster the power output, right? So in this case, the phosphocreatine system, one step, one ATP, so it's really fast. But again, the downside is you can only store so much phosphocreatine because it's too heavy. So let's move on then to the macronutrients. So the macronutrients, as I've mentioned, those are nutrients that we break their bonds in stepwise fashion, and as we do that, that is going to help us remake or rephosphorylate ATP. So you can see the 4 macronutrients there, so carbohydrates, fat, protein, and alcohol. So they have different amounts of calories or different amounts of energy per gram. So you can see if I have a gram of carbohydrates, that's 4 calories. Mine all have kcals in front of them. So if you've taken chemistry, or if you remember chemistry, so you remember that a calorie is the amount, see, I should have looked this up before class. So a calorie is the amount of heat required to raise 1 gram of water by 1 degree Celsius, I believe. So a kilocalorie is to raise the temperature of a kilogram of water by 1 degree Celsius. You might want to double check that, but I'm pretty sure that's how that breaks out. So that's, anyway, kilocalorie is more correct for the human body because actual calories are just too small of a unit of measure. So kilocalories is more correct. So in classes going forward, you'll get into more of the technical details on that, but you'll see kcal more often. So you don't see calories on labels, but they are measuring kilocalories. So anyway, that's why that's on there. So per gram of carbohydrates, 4 calories. You can see there per gram of fat. So if we have the same weight of carbohydrates and fat, fat has more than double the amount of calories. So fat is a really efficient storage form of energy. You got a lot of energy in relatively little weight. So that's kind of good and bad. It's good in terms of we can store lots of energy in relatively little space. It's bad if you're trying to deplete your subcutaneous fat stores. If you're trying to get rid of fat, well, like I said, it's an efficient energy source. So you're going to have to burn a lot of calories. So that's kind of the downside there. Protein, 4 kilocalories per gram, and alcohol, as you can see there, 7 calories per gram. So sometimes, not very often, but occasionally there will be students who are convinced that alcohol has no calories in it, that if they were to drink, so if they drink something like vodka as opposed to like with no mixer, so if they're just doing shots, that there aren't any calories in that. Again, this is a rare student, but I've encountered students both here and at a previous institution who were firmly convinced that drinking liquor had no calories, when in fact it does. So you have to break down ethanol. And we discussed that actually last time when we talked about how the liver works. Remember, so ultimately you're going to make the product into acetyl-CoA, and then you have to metabolize that in the mitochondria. So there is energy, there are calories in alcohol. So, and one of the things that you'll see, here's my little food label here from some Doritos, because they are the best chip. So here's your typical food label and what it looks like. But one of the things that you'll notice is that, so you'll have grams of fat. And so if you want to know how many calories come from fat in those Doritos, all you do is take your 8 grams, so take your weight, multiply it by 9, right? Because we got 8 grams, so 8 of these, and then you have 9 calories per gram. And so then you get 72 of those 150 calories come from fat. And if we go down here to the carbohydrates, so you have 17 grams of carbohydrates and then 4 calories per gram. So then you can multiply that out and find out how many calories come from carbohydrates. And then same thing, so 8 calories, because that's pretty easy math, coming from protein. Now, one of the things that you do if you multiply all those out, you'll notice that they don't often add up exactly to the number that's on the label. So there are some rounding issues in there, but generally it's pretty close. So if in other classes you have to figure out like proportion of calories from different sources, that's how you do that. All right, so let's get specific to carbohydrates. So in terms of their basic chemical nature, so you can see that their chemical formula there, so they are carbon, hydrogen, and oxygen. So sometimes in various notes that I use, you'll see if I have something on there that just says CHO, I'm referring to carbohydrates. Now, fats are also carbon, hydrogen, and oxygen, but because carbohydrates, as you'll see shortly, are the more important for exercise, especially for high intensity exercise, carbohydrates are usually the ones that are abbreviated CHO. So in terms of their general chemical formula, you can see it on there. So CH2O to the N being anywhere between 3 and 7. So for example, with something like glucose, the N is 6. So for example, the chemical formula for glucose is C6H12O6. So obviously you just multiply the 6 by 2 on the hydrogens there. And so that's your chemical formula for glucose, but also for fructose and galactose, as you'll see here in a second. So that's going to become somewhat important in a second when we talk about glycolysis or the breakdown of sugars. So that's their basic formula. In terms of the types of carbohydrates, so monosaccharides, mono obviously means one, so think about something like a monorail. There's only one rail. So monosaccharides are single sugars, so those include things like glucose, fructose, galactose. And then oligosaccharides oligo means a few. So oligosaccharides are typically sugars that are 2 to 10 of those monosaccharides linked together. So one of the more common forms of oligosaccharides are disaccharides. So di is 2. So disaccharides would be something like glucose and fructose bonded together, as you'll see on the next slide, or glucose and glucose bonded together, right? So those are your disaccharides. But because they're more than one, they fall into the oligosaccharide category. And then the last one on there is polysaccharide. So poly means many. So that is more than 10 up to several thousand. Now, one of the questions I have for you is, where do we get carbohydrates from? What is our primary source of them? And the answer to that question is plants. So there is some sugar if you're able to drink milk. There's some sugar in that. There's lactose in that. And there are also, there's a little bit of carbohydrate in some of the meat that you eat because you are consuming the animal's glycogen, which is its storage form of carbohydrates. But by and large, the vast majority of our carbohydrate sources come from plants. All right, so let's talk about the monosaccharides. So you can see those there. So glucose, fructose, and galactose. So things to know about them. So they all have that same chemical structure. So they're all C6H12O6, but they do have a slightly different carbon-hydrogen-oxygen linkage. And so that makes them act differently in the body, right? So they're isomers. They have the same chemical formula, but a different shape. And because they have a different shape, they act a little bit different in the body. And then another thing to know about them is that for fructose and galactose, both of those two, once we absorb them in the small intestine, well, then they're eventually going to make their way circulating to the liver, and in the liver we're going to convert those to glucose before they enter into the general circulation. So we're going to convert fructose and galactose to glucose. So glucose is going to be the key player. So when we talk about carbohydrate breakdown, we're going to talk about glycolysis, which is what happens to glucose. So again, we'll convert those other two. Other things about them. So fructose is the sweetest of them. So that's typically found in fruits and honey. So some of it moves directly from the digestive tract to the blood, but again, most of that is eventually converted in the liver, and some of that is also going to be metabolized to lactate. Galactose doesn't exist freely in nature, so it's combined with glucose in lactating animals, and then again, we're going to convert it to glucose in the liver. So the oligosaccharides, so again, for us, the disaccharides are the most important or the most relevant. So lactose, the milk sugar, you can see there is a combination of one glucose molecule and one galactose molecule. And so if somebody is lactose intolerant, what that means is they lack the enzyme, which is lactase, because ACEs are enzymes. They lack the enzyme to break that bond between glucose and galactose. And so what you get then is if somebody lacks that enzyme, you get a lot of those disaccharides in the small intestine. And so that creates an osmotic gradient. So you've got a lot of relatively large molecules there in the intestine that we can't break down. And so that is can do a couple things. One, it can simply keep you from being able to move water out of the lumen of the intestine and absorb it. Or if it's concentrated enough, it can actually pull water from the basement membrane underneath the small intestine. I can actually pull water into the lumen of the intestine. And so either way, that's going to ultimately cause gastrointestinal discomfort and diarrhea. So that's why it causes that. So that's lactose. And then sucrose is table sugar. So that one's a pretty common sugar. And so again, that's the combination of glucose and fructose. And then the last on there is our maltose. And so maltose is found in beer, breakfast cereal, and germinating seeds. And as you can see there, it's a combination of two glucose molecules. The last term on there is gluconeogenesis, which is to make new glucose. So we can make a new glucose most commonly if you are carbohydrate depleted. So for example, if you ate dinner, let's say you eat at old people hours, and you eat at, 4:30, 5 o'clock in the afternoon, and then you don't eat again until after your workout the next morning. So maybe you don't eat again until like 10, right? So if you are exercising then in that fasted state, well, you've probably already depleted the glycogen in your liver. So remember that we use liver glycogen to keep our blood glucose within normal limits. And so you may have depleted your liver glycogen, but certain types of cells, so for example, red blood cells and neurons, need glucose to run on. And so to facilitate that or to meet that need, we can make additional glucose out of things like amino acids. So amino acids are the building blocks for proteins. And so what we're going to do then is convert those amino acids in the liver into glucose. Well, where do we get those amino acids from? We get them from breaking down muscle. And so if you're exercising fasted, again, usually first thing in the morning, one of the things that you're going to do is you're going to break down muscle so that you can use the amino acids from that muscle to help you make more sugar, right? So you can, through that process of gluconeogenesis, you can make additional glucose molecules to fuel the cells that need it and to keep your blood glucose within normal limits. Again, primarily in that case for the brain, because the brain, those are neurons and they rely on glucose. But you can also make new glucose out of pyruvate and out of lactate. So pyruvate is going to be the end product of glycolysis, and I'll show you where that comes in here in a minute. So those are our single sugars, our a few sugars, and then we have our many sugars. So our plant polysaccharides. So again, a polysaccharide are more than 10 sugars bound together, right? So you have a long string of glucose molecules is a really common type of polysaccharide. So in the case of plants, plant polysaccharides really fall under two categories. So you've got starch, which, so I just talked a second ago about glycogen. So remember that glycogen is your storage form of carbohydrates. So we can store carbohydrates, we can store glucose inside of muscle cells. So that's muscle glycogen. And we can also store carbohydrates inside of liver cells, right? So that's your liver glycogen. So our storage form of carbohydrates is glycogen. And for plants, their storage form of carbohydrates is starch. So we see starch in your starchy vegetables. So in things like white potatoes, sweet potatoes, peas, carrots, those are common sites or common types of plant starches, right? So you don't have to know about amylose or amylopectin. What those relate to are the digestibility of the starch. So you can see in the lower right, I have a picture of amylose. And so amylose is these long strings of little glucose molecules. And so starches that are high in amylose break down slower, because effectively what I'll do is, or what we'll do is we'll split off one little glucose here, absorb it, break this bond, split off this glucose, absorb it, break this bond, absorb it, right? So that's a pretty slow process. It's kind of Pac-Man style. Absorb this one, absorb this one, absorb this one. As opposed to starches that are high in amylopectin are absorbed really quickly, or much more quickly than those that have a lot of amylose. And the reason for that is because they have these branching glucose molecules. And so you can break off this one, break off this one, this one, right? And so you can break off a lot more glucose in a lot less time. So the more branching there is, the more quickly we can break apart the glucose molecules. So those that are higher in amylopectin are able to be absorbed more quickly. So those are our starches. And then fiber. So not all strings of carbohydrates are created the same. So for example, the structural carbohydrate in plants is called cellulose. So cellulose makes up things like your plant stems. And obviously, if you were to eat stems of plants, you can't break those down, right? Those pass right through your digestive tract. But cellulose is a polysaccharide, right? So it's a long string of glucose. Why is it that we can't break it down? And the answer is that there's a little bit different bonding between the glucose molecules in cellulose versus something like maltose, which I'll show you a picture of here in a second. But you've got different types of bonds, so different alpha and beta bonds, and so we can only break half of those effectively. And so because of that different type of bonding, so you can see there with our maltose, so maltose, again, that's your type of sugar. It's a disaccharide that's found in beer and germinating seeds and cereals. And so maltose has this little alpha bond here, and we have the enzymes to break that down. So we can split this disaccharide into two individual little glucose molecules, which is important because we can only absorb monosaccharides. So it's important that we can split that bond, as opposed to, going back to our cellulose, you've got some beta bonds here. Bring my arrow back, there we go. These ones up here, so this one and then this one, and we don't have the enzymes to break those bonds. And so because of that type of fiber, so that is referred to as insoluble fiber, that's your cellulose, that type of fiber passes right through the digestive tract. So as you can see there's two types of fiber, soluble and insoluble fiber. The gist of it is that soluble fiber kind of gelatinizes in water. So certain, if you've ever seen someone use, or if you have used yourself, certain fiber supplements like Metamucil. So Metamucil is largely soluble fiber. So if you dump that in water and then stir it up, it kind of makes a gel out of the water, right? That's a soluble fiber. So in we find soluble fibers oftentimes like inside of certain fruits or vegetables. So for example, like the inside of an apple, that's soluble fiber, as opposed to the peel of an apple, that's insoluble fiber. So there are some differences between the two, obviously. So the insoluble fiber, we break it down a little bit because again, you can break some of those bonds, but not all of them. So the insoluble fiber largely just kind of passes through the digestive tract, as opposed to the soluble fiber is going to hang on to a lot of water. It's going to basically gelatinize inside of your digestive tract. So both of them give you increased fecal bulk is usually a way that's put. So you have increased stool output from both of them. And they both do have health benefits. So that's why my little my third little bullet is on there. So for example, high fiber intake is linked to lower incidence of obesity, lower systemic inflammation, lower incidence of hypertension, which is high blood pressure, decreased insulin resistance, and less incidence of digestive disorders, including cancers along the digestive tract, decreased blood cholesterol, and decreased incidence of heart disease. So most Americans consume somewhere between 12 and 15 grams of fiber per day, but the recommendation is markedly higher than that. So the recommendation is 38 grams for men up to 50 years old and 25 grams for women. So in terms of things that fiber does, so it's because of that fecal bulk, it helps speed transit times out of the GI tract, so it helps you be more regular. And so because of that, if there are carcinogens in some of the stuff that you've eaten, well, then they don't hang around in your large intestine quite as long. And so perhaps that's one of the reasons that they reduce the incidence of colon, or that higher fiber intake is associated with a lower incidence of colon cancer, for example. That's one theory, right? It's one idea. The soluble fiber reduces your LDL, your bad cholesterol. We'll talk more about that in the second part of this talk. And so the thought there is that it kind of binds to it. It keeps saturated fat from being absorbed and possibly keeps some cholesterol from being absorbed. So that's one of the ideas there. But one of the things that both of them do, so again, they actually speed up time. They speed up movement of food through the large intestine. But in the small intestine, they actually kind of slow down the absorption of some of the things that you've eaten, right? So they actually slow down the absorption of the macronutrients. So if you consume, let's say like a soda, right? Drink some Mountain Dew. That's basically straight simple sugars, and so those are going to be, if you're consuming that on an empty stomach, those are going to move really quickly from the stomach to the small intestine. It's easy to break those down into individual little glucose molecules and absorb that, well, glucose and fructose, and absorb those molecules really quickly into the bloodstream. And so because you can absorb them so quickly and easily, you're going to get a really big increase in blood sugar, and so then you have to release a bunch of insulin to try to bring your blood sugar back down within normal limits. And so you get these big spikes of blood sugar, big spikes of insulin. If you take some simple sugars and combine them with fiber, so in something like an apple, again, there's fiber in that, and so because of the fiber, it's not as easy for those digestive enzymes to get at those sugars, so to get at those the glucose and fructose and break them from being polysaccharides and oligosaccharides into monosaccharides. And so it just slows down absorption time, and so then you don't see the big spike in blood glucose, you don't see the big spike in insulin release. And so one of the other things there is with that whole big spike in blood sugar, big release of insulin, and so the body then responds by pulling glucose out of the blood, right? You move it into the muscle, you move it into the adipocytes, the fat cells, and then once blood glucose goes back down, well, you start to feel hungry again. And so if we can mitigate, if you will, minimize those spikes in blood glucose and spikes in insulin, then that also helps us control our calories a little bit more, helps keep feeling of satiety, feeling of fullness for a little bit longer. So that's my pitch for fiber and why it's important in both of those things to reduce both obesity and diabetes. All right. So, as mentioned, only monosaccharides can be absorbed. So the things that we eat are typically include polysaccharides, right? So you've got these long strings of sugars. And so as mentioned in chapter 23, as soon as you take them into the mouth and start chewing, remember we've got salivary amylase. And so the amylases are enzymes that break down carbohydrates. So we're going to start breaking down those carbohydrates as soon as we get them into the mouth and incorporate them with saliva. Once we get that chyme from the stomach to the small intestine, then that chyme is going to be exposed to pancreatic amylase, because remember that happens in the duodenum, so the first part of the small intestine. And so we break those polysaccharides then into oligosaccharides, so again somewhere between 2 and 10, and into individual little disaccharides, or sorry, not individual, into disaccharides, so double sugars. So then our double sugars, or our disaccharides, so you've got your lactose here if you drank some milk or had some cheese, yogurt, etc. Maltose here, and then sucrose. So then what we're going to have to do is the individual little intestinal cells, pictured here. So you've got here's an individual little intestinal cell, right? So it's the epithelium of the small intestine. And so they have brush border enzymes, which you can see pictured right there. And so those brush border enzymes are going to split that last bond. They're going to split those disaccharides into monosaccharides. So here's our little individual glucose molecule. And so once we've got monosaccharides, once we've got individual little glucose and fructose molecules, well, now we can absorb them. Whoop, back a slide. And so in the case of glucose, it's actually going to move. So you see Na+, so you see sodium here. So glucose is actually going to be co-transported with sodium. So sodium is going to move along its concentration gradient. So where my arrow is up here, this is the lumen, the opening of the intestine. And so you've got a lot more sodium out here than you have inside of our intestinal cell. And so again, sodium wants to diffuse with its concentration gradient from an area of high concentration in the lumen to low concentration in the intestinal cell. And so what we'll do then is we'll allow sodium to pull a glucose along with it. So that's your co-transport. Now that we've got glucose inside of our intestinal cell, we're actually going to actively pump the sodium back out. So you can see our little ATP over here. We're going to pump the sodium back out to make sure that that gradient stays intact, to make sure that the inside of the cell maintains a really low concentration of sodium. And then we're going to, through facilitated diffusion, because you're going to have more glucose in the cell than you have out here in the basement membrane. So this is connective tissue, and then obviously this is our little capillary. So glucose then is going to move through facilitated diffusion through this little carrier out of the cell, and then eventually make its way into the capillary, where it'll end up in the liver and in the central circulation. Fructose is transported a little bit differently, and so you can see that here. Where's that? It's over here. So glucose and galactose both are co-transported with sodium. Fructose just moves with facilitated diffusion, so it's just going to move with its concentration gradient through carrier channels. So there's our little fructose. And so again, it's going to move the same way down here. All right. So after we get those carbohydrates into the bloodstream, well, then what happens? Well, you've got three options, really. So the first is that if we are exercising, if we're moving, if we just are depleted on calories, whatever, if we need those carbohydrates for energy right now, well, we can use them for that. And so the first is that that glucose can immediately become available for energy. We can break it down and help us remake ATP right now. So an easy example of that would be if you are, we'll go big, if you're running a marathon, you're going a long way, and you're at mile 10 or 12 or whatever, and you drink some Gatorade, right? Or you eat some jelly beans or something like that. Obviously, you're running through quite a bit of ATP because you're having repeated muscle contractions. And so you got to fuel that muscle contraction. So we need the glucose right now to fuel that muscle contraction. So that's one time where that would be the case. Or if we don't need it for energy right now, then we can transform that glucose into glycogen, right? So we can store it in the liver, or we can store it in the muscles. And so you can see that depending upon the size of the person, their nutrition status, etc., their training status, those kinds of things, in general, you can store about 100 grams of glycogen in the liver. So thinking back to one of the previous slides, so if you've got 4 calories per gram, you can store about 400 calories worth of carbohydrates in the liver. And then in the muscles, again, you can store 400 grams, so quite a bit more skeletal muscle because your liver weighs about 3 pounds, so hopefully you've got quite a bit more muscle mass than 3 pounds. And so because of that, you can store a lot more glycogen in the muscles, so 400 grams times 4, so you got about 1,600 calories worth of carbohydrates stored in the muscles. And then, so between the two, you can store about 2,000 calories worth of carbohydrates, right? And so one of the ways to think about that is the ballpark figure is to walk or run a mile costs you about 100 calories. So for somebody, for like 175 pound male, that goes up a little bit. It's more like 125 calories. For somebody who's, 100 pound female, it drops a little bit below 100, but give or take, it's about 100 calories per mile. And so if you can store 2,000 calories worth of carbohydrates, you effectively have enough carbohydrates to fuel exercise or to fuel you for a 20-mile run, right? You use a similar amount of calories for walking and running. There's a little bit of a difference, but it's not a huge difference, at least during the exercise. So at any rate, you've got enough carbohydrate stores to carry you for about 20 miles. Fat, on the other hand, much more efficient fuel source, and I think I might have mentioned this little statistic in this class previously in the semester, but remember that fat, we've got 9 calories per gram, you've got subcutaneous fat stores, and then some visceral fat stores throughout the body, and so you actually store way more calories in terms of fat. So that same 175-pound guy with pretty average body fat, so something like 15% body fat, has almost enough calories to traverse the entire west coast of the U.S., right? So we can go 20 miles on carbohydrates. We can go most of the west coast on fat. So fat is a much more efficient fuel source in terms of energy density. And that 20-mile number I gave you, one of the things related to marathon running, oftentimes people will complain of or try to avoid bonking or hitting the wall or whatever term they want to use, which occurs somewhere in the neighborhood of mile 20, maybe mile 22. And so what they're describing, so usually if you ask them what that feels like, people will say, well, it just feels like, man, around mile 22, like I'm just completely exhausted and I just can't keep up, and it's really hard to keep the pace and all those sort of things. So that's depletion of glycogen. As we'll see here in a second, you get a much faster production of ATP from carbohydrates than you get from fat, basically because there's less steps involved in the process. And so because of that greater power from breaking down glucose, then when you run out of glucose, so if you're glycogen depleted, then you're having to run on fat. And so because of that, the exercise becomes much more difficult. You can't keep the same pace, those kinds of things. So avoiding that glycogen depletion is really important for long-term endurance exercise. And you can get pretty close to glycogen depletion in some of the really intense interval sports. So things like soccer, basketball, potentially lacrosse, depends on how much they play. Sorry, my phone rang. Wife was calling. So anyway, avoiding that glycogen depletion is important for being able to maintain the same intensity of exercise. And then the last thing there, if your glycogen stores are full, so you've just been, hanging out all day, so you're not using the energy right now or not using much, and your glycogen stores are already full in your liver and in your muscles, well, then we can convert that glucose to fat, and then we can use it for energy later. All right. So as you can see there, the primary function of carbohydrates is to provide energy for cellular work. So carbohydrates, we use them for other things. So for example, think way back to chapter 3, we talked about the glycocalyx, which is the sugar covering of a cell, the way that we identify cells within the body. So like the way that the immune system identifies them. So we need glucose to help us make the glycocalyx. Glucose or carbohydrates are also involved in the reproduction of nucleic acids, so think DNA, RNA. But those few things aside, the primary job, the primary use for carbohydrates is as an energy source. The second bullet point on there, so carbohydrates are the only macronutrient whose stored energy can generate ATP anaerobically. So in order for us to break down fats and proteins to remake ATP, there has to be oxygen available. In the short term, So for intense exercise for 90 seconds or less, we can break down carbohydrates to provide energy at a rate faster than we can supply oxygen to the muscle cells. So we can break down carbohydrates anaerobically, so not in the presence of oxygen. So where that becomes important is during really intense exercise. So during sprinting or slightly slower than sprinting. So if you're running something like a 400 and 800, something like that. So pretty intense exercise for 2 to 4 minutes. You're going to use carbohydrates are going to provide the vast majority of that energy. So again, they're the only ones that we can break down anaerobically. And so the way that we break carbohydrates down anaerobically is through a process called glycolysis, which I'm going to show you on the next couple slides. So this drawing isn't as complicated as it looks. So what this thing is a cell. And so this thing is the mitochondria. And so effectively it's showing you how you break down each of the macronutrients. So as we'll talk about here in a second, carbohydrates, we break them down. Eventually we end up with glucose. Glucose goes through glycolysis. We end up with pyruvate. That ends up as acetyl-CoA, go through the citric acid cycle and the electron transport chain, and then we end up with our waste products. And then, so that's the basic part of, or the basis of what we're going to talk about today. And then once you've got that down, fats and proteins are going to follow the same basic pathways. It's just a matter of where they come in along the pathways. So different amino acids come in at different points in the citric acid cycle, for example. Fatty acids, we're going to convert them to acetyl-CoA, and then they go through the citric acid cycle. So the reason we're doing carbohydrates first is if you understand this process, so straight down, and again, the three basic components we're going to talk about today are glycolysis, the citric acid cycle, and the electron transport chain. Once you've got those down, well then fats and proteins are pretty straightforward because they just come in at different points somewhere in those different cycles. All right, so let's talk about glycolysis. So hopefully this is a review. I would imagine it is, that you probably saw it at, maybe in Bio 120, or that at the very least you've seen it in some of your coursework in high school or before this. So again, glycolysis is the breakdown of glucose. It is an anaerobic process, meaning that oxygen is not going to be involved here. So we'll start up top. So this is our little individual glucose molecule. So glucose is a 6-carbon ring, hence the little shape here. And so the first thing that we're going to do is we are going to import glucose inside of a cell. And so once we do that, so we're going to pretend that there is a sarcolemma here where it says glucose. So that's our muscle cell membrane. So we're going to import glucose inside of the muscle cell, and once we do that, we're actually going to phosphorylate it. So we're going to add a phosphorus to that glucose molecule to give us glucose 6-phosphate. So as you've probably noticed, we had to give up energy as soon as glucose got into the cell. Once we've transformed glucose into this glucose 6-phosphate, it can either continue along this pathway, as we're going to do in a second, and it can produce energy for us, produce ATP for us, or if we don't need energy right now, then what we can do is make it into glycogen. So we can add that glucose onto a string of glycogen inside of the muscle cell, and so then we have our stored carbohydrates. But we're going to continue on down the pathway. So a few things about this diagram. The numbered things are the enzymes that cause that step. So hexokinase is going to be involved in phosphorylating. So again, adding the phosphorus group to glucose. Phosphorylase is the enzyme that's going to be involved in glycogen production. And then, so anything numbered, again, those are the enzymes. Anything highlighted there, those are your rate-limiting enzymes. So they play a role in how quickly this whole process can continue or not. So we take our little phosphorylated glucose, another enzyme here. And so you don't have to know any of these enzymes. I'm kind of just, what I want to do is outline the process for you, and then I'll talk about why it matters here in a second. So got another enzyme here. We change the shape of the molecule, so we end up with a fructose 6-phosphate. We phosphorylate it again. So we actually break down another ATP, so we have another energy input into this process. And so the product at the end of step 3 is, you can see, fructose 1,6-diphosphate. And so what matters here, so you've got a phosphorus here, or a phosphate here, and there as well. And so why that matters, so you've got your 6-carbon ring, two phosphates on it. And what we're going to do the next step is actually split it in half. So then we're going to end up with two phosphorylated 3-carbon fragments. So let's do that. And so that's step 4. And so you can see then that we have 2 pathways, and all that represents is that we split that single 6-carbon molecule into 2 3-carbon fragments. So we got a 3-carbon fragment going left and a 3-carbon fragment going right. So step 5, change it again. And so what you notice here at step 6, so remember that in the intro part of this, I said that digestion, or the energy production process, we're going to essentially pull hydrogens off of the macronutrients, and then we're going to use those to help us remake ATP. Well, this is the first time that we can see that. So there's a couple different things to talk about, and they're included on a later slide, about 2 slides down. But the concepts of oxidation and reduction. So, and again, these are on in a few slides. But oxidation is giving up electrons. So in this case, this 3-phosphoglyceraldehyde gave up hydrogen, right? So it gave up electrons. So 3-phosphoglyceraldehyde has been then oxidized. It gave up electrons. If we reduce something, then that means to accept electrons. And so NAD has been reduced. It accepted that hydrogen. And so it's actually going to take this hydrogen into the mitochondria. To get rid of it, we're going to attach that hydrogen to oxygen to make metabolic water, so to get rid of those hydrogens. So we need these coenzymes. So NAD, I won't go into the full name because I'm just going to refer to them as NAD. And then there's one involved in the citric acid cycle, which is FAD. So both of those coenzymes come from B vitamins. So I mentioned way earlier the concept of macronutrients. So again, carbohydrates, fat, and protein, and that we're going to break the energy in those bonds, or we're going to break their bonds and use the energy in them to help us remake ATP. So those macronutrients are as opposed to micronutrients. And so micronutrients are things like your vitamins and minerals. So we talked about vitamins a little bit previously. So A, D, E, and K being the fat-soluble vitamins. Well, in this case, we're talking about some of the water-soluble ones. We're talking about the B vitamins. And so sometimes people talk about B vitamins as giving you energy. And so they don't directly give you energy, but they are involved in the energy production process. So again, niacin is a B vitamin. So if you were deficient in niacin, well, you wouldn't be able to make enough coenzymes. You wouldn't have enough NAD, and that would, theoretically, hamper your ability to break down glucose, right? And so by having sufficient intake of the B vitamins, of niacin and riboflavin, so FAD is a derivative of riboflavin, by having enough of those B vitamins then, that enables you to carry out glycolysis, and then again, riboflavin is involved in the citric acid cycle, so it enables you to carry out macronutrient breakdown. So in the absence of the micronutrients, so again, particularly the B vitamins, then that hampers your ability to break down the macronutrients. So that's why, so for example, on energy drinks, a lot of times, like Red Bull and various others, you'll see really high doses of B vitamins. That's part of the rationale, is that you need some of those B vitamins to help in the energy production process. So there's that. All right, so at step 6 here then, again, so we are going to reduce NAD, so it's going to moved my mouse too far. It's going to accept a hydrogen here, and again, it's going to take it into the mitochondria. Step 7, finally. So seven steps in, we finally get back an ATP. So we finally gotten some energy back. The only problem, of course, is that we had to put 2 ATPs in on the front end. So we get an ATP on this side, get an ATP on this side, but now we're basically just back to 0 because we put 2 ATPs in to start. We got 2 ATPs back, so we're back to 0. In terms of energy balance, step 8, step 9, step 10, we finally end up with 2 additional ATPs. So at the end of glycolysis, you end up with 2 ATPs made anaerobically, so no oxygen involved in this process. So with 10 steps, you can make 2 ATPs. So it's a pretty quick process, but think back to phosphocreatine. Remember, so that was 1 step, 1 ATP. This is 10 steps, 2 ATPs, right? So it's pretty quick, but it's still not nearly as fast as we get with the phosphocreatine system. And so because of that, the energy output is lower. So after you go all out for 10 seconds, you're going to start to slow down. And the reason you're going to start to slow down is because you've depleted your phosphocreatine and you've essentially switched over to the glycolytic system. Now, the body doesn't exactly shift gears like that, but that's kind of how it works. It's an easy way to conceptualize it. So in products then of glycolysis, 2 ATPs, or 2 net ATPs, we also then end up with 2 reduced coenzymes, so 2 NADHs, which will take over inside of the mitochondria. And then we end up with a pyruvate molecule, so actually 2 pyruvate molecules, so 1 on each side. So one thing I need to tell you before I forget is that, so again, this is happening inside of the muscle cell. So all of the anaerobic stuff, so all the stuff not requiring oxygen, happens in the sarcoplasm. So remember, that's the cytoplasm of the muscle cell. So glycolysis, all these enzymes are in the sarcoplasm of the muscle cell. So everything else, everything aerobic, is going to happen inside of the mitochondria. So the phosphocreatine system is in the the cytosol, but in specific to the muscle cell, that's the sarcoplasm. And then glycolysis is also in the sarcoplasm. So we end up with our pyruvate, and I'll actually come back to lactate in a minute, and we'll talk about what happens, why it's going to be formed. So we have our 2 pyruvates. So let's say we are exercising at a relatively low intensity, right? So we are walking or maybe slow jog, something like that. So then what's going to happen is why I mentioned that lower intensity is because it's relatively easy for the body to supply enough oxygen to meet the demands of that exercise. So it's easy for the heart to pump the blood around and deliver enough oxygen to the muscle cells so that we can use that oxygen to remake ATP. So we've made these 2 anaerobically, and then we're going to take our pyruvate and we are going to transport it inside of the mitochondria. So here's my muscle cell, just in case you forgot what they look like. So you got your contractile proteins here. So all this space out here, that's our sarcoplasm, and then here's our little mitochondria. So we are going to transport our pyruvate inside of the mitochondria. Once it gets inside there, we reduce another NAD to make an NADH. So first step here, we're going to convert that pyruvate to acetyl-CoA. So that's a little 2-carbon molecule. And then we're actually going to join it to a 4-carbon molecule over here, oxaloacetate. And so we combine those 2 and we end up with a 6-carbon molecule. And so then we have another 10, so 11 total, but another 10 enzymes involved here. And so we go around the circle. And so through the course of those 10 enzymes, you're going to see that we're going to reduce another NAD. And then we go another couple steps, reduce another NAD. We get another ATP here, reduce an FAD, and then reduce another NAD. And so in the citric acid cycle, so this is going to happen again inside of the mitochondria. For every time we go around that cycle, we're going to end up then with 1 ATP that we've made, 4 molecules of reduced enzymes, so 3 NADHs and 1 FADH2, and then we're going to end up with 2 carbon dioxides. And because each glucose molecule yields 2 pyruvates, well then just double those numbers. So for each molecule of glucose, you get 2 carbon dioxides, 8 molecules of reduced enzymes, and then 2 ATPs. So as you can tell here from the citric acid cycle, we only got 1 ATP back. So where does the energy production come in? Where am I making these ATPs? And the answer to that question is the third part of the process, which is the electron transport chain. So that's where we're really going to get our ATPs back. So what you're looking at here, so as you can tell from the drawing, so this is a mitochondria. So again, what we just talked about, the citric acid cycle happens inside of the mitochondria here. So remember that the mitochondria has 2 layers to the membrane. So there's the outer mitochondrial membrane here, but also here. And then, bring that back. And then there's the inner part of the matrix here. So the citric acid cycle happens on the innermost part, so inside the inner matrix. So what you're looking at down here, this is inside of the mitochondrial matrix. This is that space between them. And then this is outside of the mitochondria. So this would be the sarcoplasm. So this is inside of the muscle cell, but outside of the mitochondria. So I mentioned oxidation or reduction. So again, oxidation, giving up electrons, reduction, accepting electrons. And so what's going to happen is with all of those NADHs, all of those NADs that have been reduced, that have accepted hydrogens, they are going to move to this cytochrome complex. So there are 4 iron-containing cytochromes. So you can see the Roman numerals there, 1, 2, 3, 4. And so what's going to happen is that NADH is going to be oxidized. So it's going to give up its hydrogen. And so actually what we're going to do is we're going to pull some electrons off of the hydrogen. So we're going to split an electron there. And so then, trying to bring my mouse back, there we go. So cytochrome 1 is going to be reduced. So it's going to accept electrons. And then we're going to transfer those electrons from cytochrome 1 to coenzyme Q. And when we do that, so the process of being reduced, of accepting electrons and then giving them up, that actually gives off a little bit of energy. And so when we give off that energy between cytochrome 1 and coenzyme Q, that energy that's given off we use to basically kick out or pump out, if you will, hydrogen into this intermembrane space. So we're kicking hydrogen out here, and then those electrons move from coenzyme Q to the 3rd cytochrome. So again, coenzyme Q is going to be oxidized, so it's going to give up its electrons. Trying to bring my mouse back again, sorry. So it's going to be oxidized, it's going to give up its electrons, so then we're going to reduce cytochrome 3. Then it's going to give up its electrons to cytochrome C here. And so again, when we do that, when we transfer the electrons from cytochrome 3 to cytochrome C, that is going to give off a little bit of energy. And when we do that, that's going to pump another hydrogen out. Same thing here at 4, pump another hydrogen out. And so what we're trying to do is steal energy from those electrons, because you've got high energy electrons with a lot of energy out here, to progressively less energy as we oxidize, or sorry, as we reduce and oxidize. So as we do that, the electrons are going to progressively lose energy, and as they give off that energy, we're going to use that to pump hydrogen out here. So we end up with a whole lot of hydrogen in this intermembrane space. And so that then creates an electrochemical gradient, right? So you've got lots and lots of hydrogens out here in this intermembrane space, not very much inside. And so because you've got lots of positive ions out here, not as much in here, the hydrogen is going to want to move back inside. And we'll let it do that, but the way that it comes through is through these proteins called ATP synthase. So as we let hydrogen move with its concentration gradient from the intermembrane space back inside of the mitochondria, that's going to cause ATP synthase to spin. And when it does that, it's going to rejoin adenosine diphosphate and our little free phosphate here to remake ATP. So once we've created that gradient and allowed hydrogen to move back inside of the innermost membrane there, inside of the mitochondrial matrix, that again is going to spin ATP synthase and remake ATP. And then once the hydrogens are back in here, then we'll attach them to oxygen, and then we get metabolic water. So we've made water there. So tying it all together, remember one of the things we've talked about a lot in this class, thinking back to the lung chapter, for example, we talked about being able to pick up oxygen, because cells need oxygen in order to be able to survive. And the reason for that is because they need oxygen to remake ATP. And the real reason, so if breakdown of the macronutrients is essentially pulling off hydrogens, remember we got to get rid of those hydrogens somehow. And so we do that by attaching those hydrogens to oxygen and making metabolic water. So we have to be able to supply oxygen to cells so that they can have a way to get rid of the hydrogens that they strip off of the macronutrients, so the hydrogens that they strip off as part of the energy production process. So that's where oxygen comes into this. And we also talked about carbon dioxide. So we'll actually back up a slide. And so carbon dioxide, as you can tell, is going to be produced as part of the citric acid cycle. So that's where the waste products then of respiration occur. So remember, We talked about ventilation and respiration. When you breathe out, you're going to breathe out carbon dioxide. That's where we make it, is in the citric acid cycle. And then we're going to use the oxygen that we breathed in as part of the electron transport chain to help us get rid of those hydrogens that we've stripped off of the macronutrients. So once we go through all three of those processes then, and there's a little close-up of ATP synthase. So once we go through all those processes then with our glucose, we get back 32 ATPs per molecule of glucose. So remember, we made 2 of those anaerobically, but the vast majority of those we made aerobically. So we made the vast majority of those, the other 30, with the electron transport chain. All right, I do need to back up a little bit, back to glycolysis. All right, so where does lactate come into all this? So the end product again of glycolysis is pyruvate. What happens if I am, let's say, running a 400, running an 800, right? So I'm exercising pretty intensely. I'm burning through ATP faster than I can deliver oxygen to the muscle cell. So I need to be able to, at least in the short term, provide as much energy as possible. So I can take advantage of the fact that glycolysis is an anaerobic process. So what we'll do then is remember that to get rid of these hydrogens, once NAD has been reduced, to get rid of these hydrogens, this, again, all this glycolysis is happening out in the cytosol, so NADH has to go to the mitochondria, has to move inside has to move through the mitochondrial membrane, has to drop off its hydrogen at the electron transport chain, and so that's going to take time. That's a slow process. And so if I am doing some kind of extended sprint, well, I don't have that kind of time. And so what I can do to continue to produce ATP in the short term is I can take these hydrogens that NADH has picked up, or NAD has picked up to become NADH, and I can drop that hydrogen, trying to bring my arrow back, there we go, I can drop that hydrogen onto pyruvate, which is what then makes it lactate. And so once we've dropped that hydrogen onto pyruvate and made lactate, now that NAD can go back up here to step 6 and keep doing that. Once, if the NADs become saturated and they're not able to carry out this process, well, then we're stuck. We can't produce any more energy through glycolysis. So it's important that they be able to offload these hydrogens onto pyruvate here to make lactate. So lactate is kind of a temporary storage spot for those hydrogens. And so lactate then, a few things can happen to it. It can stay inside of the muscle cell, and then when oxygen becomes available, we can pull the hydrogen back off and then take it to the electron transport chain, or we can kick lactate out of the muscle cell, and then it can be used by other tissues. So for example, the heart, muscles that aren't working as intensely. So if you're running, some of the upper body musculature can metabolize that lactate. The liver can convert it back into this glucose 6-phosphate. So a bunch of different things can happen to it. But basically, if we export it outside of the muscle cell, it's going to be used for energy in a cell somewhere else. So again, usually or oftentimes muscle cells that aren't working as hard. So lactate, oftentimes referred to as lactic acid, that's made as a byproduct of breaking down glucose in the absence of oxygen. Because again, I don't have time to drop these hydrogens off of the electron transport chain and to attach them to oxygen, because I'm limited by my ability to transport oxygen from the bloodstream into the muscle cell, and from inside of the muscle cell inside of the mitochondria, right? There's a lot of steps in that process. It's not all that fast. And so because I'm limited by the speed of that, again, if I want to continue to produce ATP because I'm sprinting, we can just temporarily drop them on pyruvate. So that's where lactate comes into this process. All right. So in terms of the key points there, so energy from most cellular processes, including muscle contraction, is supplied by the breakdown of ATP. If energy production past a second or two requires rapid reproduction of ATP from adenosine diphosphate and free phosphate, we can do that with or without oxygen. So the two processes to do without oxygen are the phosphocreatine pathway or glycolysis. And then the aerobic pathway includes the citric acid cycle and combined with the electron transport chain. All right, so talked about this stuff. So the anaerobic sources of ATP rephosphorylation, so again, remaking ATP from adenosine diphosphate, the free phosphate, include the phosphocreatine and the glycolytic system. So again, for the phosphocreatine system, one-step reaction catalyzed by creatine kinase. So we can remake a new ATP in one step, so it's really quick, but it's limited by our supplies of phosphocreatine. In terms of glycolysis, so it's relatively fast, right? So it's 10 steps, and we get back 2 ATPs. So more steps than phosphocreatine, so it's not quite as fast, meaning you can't keep up quite the same intensity. So again, if we're thinking of somebody running around a track, they can go really fast for the first 100, and then they're going to slow down for that second part. For if they're running something like a 200, they're going to slow down for the second part, depending on how good they are at pacing themselves. In part because, so if we just ask them to go all out, they're going to slow down after that first 100 meters because they've depleted their phosphocreatine, and now they're just relying on the glycolytic system. So because it's 10 steps as opposed to 1, can't produce energy as fast, so we can't keep up that same power output, so we can't run as fast. Then once we get into the aerobic systems, so if we add in the citric acid cycle, well, now that's 11 more steps, plus transporting pyruvate inside of the mitochondria. That takes some time. And then we have to take all those reduced coenzymes, so the NADHs and FADHs, and drop those hydrogens at the electron transport chain, go through the 4 cytochromes. Obviously, that takes some time too. So the aerobic energy production process has much lower power, if you will, right? So we can't produce ATP nearly as quickly, and so if we're relying on the aerobic process, it's just slower. Can't keep up the same intensity. So there's our complete oxidation of glucose. So we go first through glycolysis, those 10 steps, then through the 11 steps of the citric acid cycle, then through the cytochromes, and eventually we get back a total of 30 ATPs. Again, 2 anaerobically from glycolysis, and then the remaining 30 from the electron transport chain. And so then the last thing I want you to know for this one is that carbohydrates are the fuel of choice during high intensity exercise. So we're actually going to start on the picture on the lower right here. And so what you've got, so your VO2 max is your aerobic capacity, which is your ability to deliver and use oxygen in the muscle cells. So a rough estimate of your VO2 max is your fastest mile time. Right, so if you go any faster than that, so let's say you can run a mile in 6 minutes. So if you run at a pace any faster than that, now you're starting to really incorporate the anaerobic side of things. And if you run slower than that, the slower you run, the more aerobic you become. So if we've got this person who, again, let's say their fastest mile is a 6-minute mile time, so that's 100% of their VO2 max. So let's say so if you run a 6-minute mile, it's 10 miles an hour. I did that to make the math a little easier here. So let's say they're operating at 25% of their VO2 max. So they're going at about 2.5 miles an hour, right? That's a pretty leisurely walk. So if they're engaged in this leisurely walk, the sources of energy fueling muscle contraction for that. So you can see you've got plasma glucose, so glucose from the bloodstream, plasma free fatty acids, so fat, and then muscle triacylglycerols, which is, so I stole this from an ex-phys textbook, it's triglycerides, right? So you got basically the vast majority of the energy that the primary fuel source that you're using to rephosphorylate your ATP is fat. So it's either fat from the bloodstream, so your plasma-free fatty acids, or fat that's stored inside of the muscle cells. Well, if we switch from a leisurely walk to a sort of either a fast jog or slow run, so 6.5 miles an hour here. So, that's a little over 9-minute miles. So if our person runs 6-minute miles, it's not going to be that hard for them. So when we go a little bit more intense, again, because the energy transfer from carbohydrates is faster than it is from fat or protein, because there's just fewer steps involved in the process. Well, now our energy sources for that, so we because obviously the energy source overall we need more of, right? So you got more plasma glucose, so more sugar from the bloodstream. You got your plasma-free fatty acids here, and then you've got your intramuscular triglycerides there. And then now you're using muscle glycogen. Then during pretty intense exercise, so 85% of your VO2 max, so our person who runs a 6-minute mile is now running somewhere around a 7-minute mile, so not as fast as they can go, but still pretty fast for them. They are using, the rest of the energy comes from intramuscular glycogen. So All of that is to say that the more intense the exercise, the more you rely on carbohydrates. The lower intensity of the exercise, the more you use fat as a fuel source. So at rest, if you're just sitting in a chair like I am doing this talk, you are using fat as your preferred fuel. And then the higher the intensity, the more you shift from fat as the fuel source to carbohydrates as the fuel source. So carbohydrates are the fuel of choice during high intensity exercise. And so what this picture is on the left, so you've got two groups on a stationary bicycle. And so the two groups, and this leaves out the control group, but these are the two experimental groups. So the red group, the carbohydrate depleted group, remember I said CHO is usually the abbreviation for carbohydrate. So this is carbohydrate depleted. So this group had a diet that had less than 5% of their calories come from carbohydrates for 3 days before this bicycle test. And then the carbohydrate loaded group, 85% of their calories came from carbohydrates for the 3 days leading up to the bicycle test. And they stuck both of those groups on a bike at 70% of their aerobic capacity. So vigorous exercise. So working pretty hard, certainly not all out, but it's something that you can maintain for quite a while normally. And so the group that is carbohydrate loaded was able to actually maintain a pace a little above 70% of their aerobic capacity for 2 solid hours, right? So they're able to maintain a fairly high intensity of exercise for 2 hours. The group that was carbohydrate depleted, you can see they start to fall off pretty quick. So again, to maintain this intensity of exercise, because we're thinking about 70% of the VO2 max, their bar would end somewhere around here. But carbohydrates would form a major contributor to exercise at that intensity. And if you don't have carbohydrates, well, then you just can't keep up the intensity. And so because of that, because they only have fat and protein to use, because they're carbohydrate depleted, they just can't keep up the intensity, and so their performance starts to fall off, so that by the end of that 2 hours, they're at about 35% of their aerobic capacity, as opposed to this group is still cruising along at 70%. So as far as high intensity exercise goes, so things like sprinting, basketball, baseball, lacrosse, soccer, all those kinds of things, carbohydrates are still king, and so carbohydrates are going to be the fuel of choice during those exercises. So that's what I have for you for this first part. Like I said, the second part of the talk is fat and protein. And again, if you've figured out enough of the citric acid cycle and the electron transport chain, well then fat and protein are easy. So we'll see you next time.